Hey everybody, thanks for joining us on Skype a Scientist Live today. We're going to be tackling the situation that we're in right now, talking about COVID. We've got two awesome scientists with us today from Rockefeller in New York. So I'm just going to hand it over to them. But before I do, I'd like to remind everybody that we are a nonprofit organization that totally relies on donations from our audience. So if you can support us, it helps us put out all the content that you see. And so if you can do that at patreon.com/SkypeAScientist, those monthly donations, like even if they're really small, can really help keep us stable and kind of help us predict how much resources we'll have to put stuff out in the future. We also appreciate one-time donations too, just as much. You can do that at paypal.me/SkypeAScientist. And all of those donations are tax deductible because we're a 501c3 nonprofit organization. So we're so glad that you're here with us today. A couple ground rules. One, please be respectful in the comments. I will kick you out if you're not respectful. Please only submit your questions once, because even if you give it to me 5 times or 1 time, I'll see it, I'll read it the same amount. So if you submit your questions more than twice, I will kick you out, just because it's hard for me to find the other questions. So please be respectful. And with that, I think that's it. So I'll hand it over to you two.
Great. Thanks, Sarah. And thank you, Erin, for your work here too. And so I'm really pleased to be here as part of the Skype a Scientist endeavors, which are amazing. And I hope everybody contributes to the Patreon. So I am a scientist from Rockefeller University, and my role there is as director of RockEDU Science Outreach. This is a program that is very similar to the things that Skype a Scientist is doing, where we work to bridge the K-12 communities with the research communities and sort of create a better ecosystem around science education and the sharing of science knowledge. I'm really excited to be here today. I am sheltering from my home in the Bronx, New York. We are certainly in the epicenter of the pandemic here in the United States. And so, you know, there's a lot of sort of emotion around this as well. A lot of our, we work in hospital settings and research settings that are being bombarded with COVID stuff. And we, have a lot of friends who are doctors and nurses who are on the front lines. And I just want to give a shout out to their work right now because it's just incredible what they're doing. And I'm sheltering in place with my children. I have 2 middle school aged daughters, my dog Willow, my cat Lucy, who's 18 years old, and my husband Jim, who's also a science teacher. And so all of those things are taking place as I am presenting to you. So if you hear somebody go into the kitchen for a dish or whatever, this is just where we are. I can't keep the noise from happening, but hopefully it'll be fine for you. And so I'm really glad to be here with Gabby, and
Well, hi everyone. My name's Gabrielle Paniccia. I am a graduate student at Rockefeller University. And my area of interest is very much in how viruses evolve. And so, although I'm very early in my PhD career, my interest really focuses on viruses that infect a really wide range of animals, going from birds to people to mosquitoes. And so that's why the whole coronavirus situation has really gotten my attention and why I've been doing a lot of reading into it from home.
Cool. So I think if it's good with everybody, we're just going to jump right in. Gabby and I are going to talk a little bit about some general virus foundations, just for a few minutes to make sure that we're all up to speed. And then we'll start taking questions. I want to just emphasize, in addition to some of the ground rules that Sarah set, that we are PhD research scientists or training for PhDs, that we are not medical clinicians or physicians. And so none of this information is going to be around medical advice. It's going to be about pure just scientific knowledge around the viruses. So I just want to make that clear that we are not we're not real doctors, as my mom calls it. We're PhD doctors. So, okay. So viruses are very small molecular systems, right, that require a host organism to function. And viruses are ridiculously small. And I think I'm going to be able to share my screen real quick. But before I do, I want to point out this right here. I don't know if you can kind of see what this is. Sorry, this is a coffee bean, okay? And so you can see the size of the coffee bean sort of in my hands right now and understand what the coffee bean size is compared to me. I'm only willing to provide you with one of my coffee beans because, you know, who knows what the stash of coffee is going to be like in the future. And so now I'm going to share my screen because I want to show you this really cool resource that has been, oh, Sarah, would you mind letting me share my screen?
Yeah, so I just made you a co-host, but before you do, we just want to make sure that Erin has a chance to sign to folks that if they want to continue seeing Erin, they have to look at the top right of their screen, and there are different view options. So when you go to share your screen, I think the default is to only show the speaker, and we want to make sure that they can also see Erin. And so to do that, you should click the button that has two little rectangles on top of each other. And then once Erin's hand stop moving, you can go to screen share.
Oh, me. Oh, sorry. Did I do that wrong? Okay. You're muted. All right. Well, okay, we're good. All right, so here is that coffee bean that I showed you, right? And I'm going to take you through this amazing resource from Learn Genetics at the University of Utah that sort of gives you just helps to put into perspective how tiny these things are, right? And so we're moving in, we have the Times regular, this is like your newspaper print, a grain of rice, a sesame seed. And as we move through this, right, we are going into smaller and smaller scales. We see what the grain of salt looks like. Here are your single-celled organisms like the amoeba or a paramecium. Here is the approximate size, and this is 130 micrometers of a human egg, which is the largest cell in the human body. You can potentially see that with your naked eye. But other than that, you need a microscope. Right, and we're moving in. This is about the size of a skin cell. Then even smaller than a skin cell is a red blood cell. This is approximately the size of the X chromosome. And now we're getting into some other single-celled organisms like the E. coli. So this is the size of a bacteria. Bacteria are actually a lot bigger than viruses. And these are the things that are the size, these are things that are inside of our cells. So you might have heard of something called a mitochondria, which is like the powerhouse of the cell. So bacteria, viruses are not even as big as those cell components. And then we get in and we finally see how large or how small a virus is, right? So we see a measles virus, influenza virus, HIV viruses. These are super, super tiny. There are actually millions and millions of viruses on planet Earth. If you were to, for instance, this is a phage. A phage is a special type of virus that infects bacteria only. And if you were going to take all of the phages on planet Earth and weigh how much that is, it's actually 1,000 times the amount of weight by all of the elephants on Earth. That's how much phage we have on this planet. And that's only one specific kind of general family of viruses. There's viruses that infect plants. There's viruses that infect humans. There's viruses that infect fish. There's basically a virus for every living organism out there. And so I just wanted to sort of share that about viruses and the sizes of our viruses. Okay, so I'm gonna stop the screen share and just sort of talk a little bit more about, actually I'm gonna reshare because I have one more, I think, good visual that could give a little bit more information and help with foundations. So this is, every virus has a specific amount of kind of components that are associated with it. Okay, and so every virus, again, we are talking about a virus is infectious, right? It's also intracellular, which means it goes inside of your cells, and it's a parasite because it hijacks all of the material of our cells so that it can make copies of itself, right? And on the base level, a virus is made up of some genetic material, okay? So this could be something you might have heard of DNA. DNA is something in all of our cells, and it's our genetic blueprint that helps us to basically we get DNA from our parents, and that sort of tells us what we should look like. But in a virus, it can be DNA, or it can be another type of nucleic acid or genetic material called RNA, right? All viruses also have a little bit of a protein coat that we call a capsid, right? And this is basically there to protect the genetic material so that it is remaining intact. And then sometimes, as in the case of COVID or SARS-CoV-2, which is the virus that causes COVID, you can sometimes have a lipid envelope or a layer of fat around it that adds another layer of protection and also helps it to get into some cells. So it does have a bit of a function. Okay. All right, so stopping to share my screen here for a second, and just go through a little bit more. As I had mentioned, a virus is a parasite, and it exists to perform one single overall function, and that is to transmit copies of their genetic material, so that little squiggly red line that I showed, into a host cell. And a host cell, as I had mentioned, could be anything from bacteria to plants to animals to birds to reptiles. But in our case, SARS-CoV-2 is infecting our cells, right? And it basically binds onto the host cell, and it injects its genetic material into our host cells, and all of the little machines that are working inside of our host cells sees that genetic material, doesn't recognize that it's not human genetic material, and just starts going to work on it and making tons of copies of that genetic material and all of the proteins that need to be combined to create a virus particle. And this is how the viruses hijack our cells, right? And they need to infect our cells because viruses themselves do not possess the ability to read their own genetic instructions so that they can make and assemble more copies of themselves, right? And where did viruses come from in general? While it's really hard to study how viruses emerged on our planet, especially since they're super hard to detect, right? You can't really look at a fossil record, and they're so very small. You can barely even see them with a light microscope. We actually have to use special microscopes that are like shooting beams of electrons, right? And when we think about their origins, they emerged during a time when the earth looked super different, right? Like there was like fireballs and gaseous things and oceans everywhere. And so there is some evidence based on genetics, but genetics is a little tough in viruses, that emerged before even the first cells started to evolve on planet Earth. And then some theories also suggest that maybe viruses evolved alongside of the first cells. But whatever camp people are in, there is no denying the fact that viruses are like billions of years old. Like they are so perfect molecular machines. It's actually kind of amazing. And I guess, there are so many viruses out there that most of them are just not impacting humans. But in the case that we're in right now, we are in a situation where humans are facing this brand new strain of virus, the SARS-CoV-2. And we're learning so much about this right now. Science is learning and communicating that knowledge as we go. And so we hope that I'm gonna kick it over to Gabby in a second, who's gonna go into a little bit more specific about the coronavirus and specifically SARS-CoV-2. And then we're gonna have an opportunity for you to ask questions. But please know that just as we know only what we know, there's a lot of stuff that we still don't know, and we're gonna do our best to answer your questions. So Gabby, do you wanna talk a little bit more about coronaviruses in general?
Yeah, sure. So what is a coronavirus? So the one that we're all sheltering inside from right now is not the first coronavirus. There is a huge family of viruses that are all named for the spiky crown, corona essentially, that's on its outside surface. And so the crown is made up of a bunch of proteins that help it bind to the host cell that it's going to enter and then help it get inside. It essentially tricks the cell into eating it. Inside the coronavirus, it relies on getting its RNA genome out, so it doesn't use DNA. It uses a slightly different version called RNA. That essentially means that it's ready to go the minute it gets into a cell, and all that RNA can be turned into protein. DNA has to normally in a cell be translated first into RNA and then in protein. Coronavirus skips a step, like a lot of other viruses, just so it can move pretty fast. You've probably heard a lot of different names for it. SARS-CoV-2 is the name of the virus itself, and COVID-19 is the name of the disease that it causes. It's not to be confused with a much, much older virus called SARS-CoV. They don't say one, I think they just say SARS. That happened in 2003, which is, I believe, now extinct. We got rid of all of it, essentially, except for what exists now in laboratories, frozen down really solid, that we use for experiments. There are a lot of different species, essentially, of coronaviruses, and there are some that infect people. So this is not the first coronavirus to ever infect people. And actually, for the most part, you guys have all probably encountered one before in your life. It causes a version of the common cold. So most people have, our bodies have seen at least one type of coronavirus before. In bats, they also have coronaviruses, and it turns out that they have a lot of them, but it doesn't necessarily seem to make them sick. They just happen to carry it, which is how it seems to have gotten to us. So it essentially was probably like a bat common cold, and then through interactions with people in the wild, basically, it started realizing that it could infect people, which eventually led to sort of the precursor virus for SARS-CoV-2 getting into a city on an infected animal. Let's see, I think that's, so I do want to point out, I've been asked in the past why it spread so fast. It's not the most contagious virus on the world, which I think is kind of cool to know. But it happens to be a sort of all of the stars aligned situation. New viruses could possibly, but this one had a good chance of, essentially making it all across the world because it popped up in a city, and it just had all of the right symptoms where it wasn't too bad for people, so people tried to still go out like you normally would if you probably were recovering from the flu. And so it sort of took advantage of more of our social mechanisms with each other and the way that we interact with other people, as opposed to being of itself some sort of super virus. I think, Jeannie, I think that covers about what our normal spiel is.
Our normal spiel. So should we, Sarah, do you want to choose the questions that we take, or do you want us to just take them?
So typically I'll give you the questions out loud just so that you don't have to spend the time like scrolling through and picking out the questions, just to make it flow easier. So that's why I'm here. Great. So here's a question. You showed in your slides that there are some viruses that just have the protein coat, and that SARS-CoV-2 has the protein coat and then a lipid coat around it. And so we know that hand washing basically breaks up that lipid layer and then kills the virus. Why does it need the lipid to stay alive? Like, if you break the lipid, why doesn't just the little protein coat like make it work?
Do you want me to take this one, or?
Yeah, that'd be great. Cool. Yeah.
So viruses sort of have a very specific process, sort of like a flow chart of responses. And if you break one of the links in that chain, for the most part, they become useless. So what happens when coronavirus loses that envelope, it also would lose all of the proteins attached to it that it's required to trick a cell into eating it. When it gets eaten by a cell, let's say this one doesn't have the lipid envelope anyway, and it somehow still gets eaten. The cell puts it into a bubble, and it's a little bit like a stomach. The cell makes it really acid, and it's trying to digest it, but the virus knows to expect this. And so what that does is it actually changes the shape of the spikes, which let's just assume that this one still has, and it pulls the virus close to the outside of that bubble, and then it fuses the fatty membrane of the virus to the fatty membrane that the cell put it in. And that lets it kick out the protein parts and its RNA and get into the cell. And so without that little capsid, that essentially protein part, would never be able to get out into the cell because it couldn't escape the bubble.
Sounds good. So I'm just going to warn you, I don't know what you know. So if you don't know the answer, just say, I don't know, and then we'll keep them rolling. Cool. Okay. So does the virus get killed with heat?
Yeah, I mean, if you heat things up high enough, it will impact the integrity of that lipid membrane. And as Gabby said, it will break the chain. Heat also leads to the proteins that are associated with the virus to like kind of like jumble jamble and mush up, and it will impact how that the proteins work. So I think when people think about sterilizing things, there's a couple of ways that you can do it. Like your dishwasher has like the steam setting that will probably, that could sterilize your dishes and stuff like that. Not to say that coronavirus would be on your dishes, but like, that kind of temperatures. And then you can also use things that are not heat related, right? So we know that alcohol can keep the virus from sort of being a viable thing. So a lot of people are wiping down their doorknobs, and like, I know we live in buildings, so like elevator buttons and stuff like that. And hand sanitizer could also be helpful, but ultimately washing your hands is the best way to kill the virus from you.
Sounds good. Let's see. Can you get immunity to the coronavirus?
Yes. So we don't advise people going out and getting sick with it in order to get immunity, because that's not going to help the situation. But essentially, it seems like people who were originally infected with it are then immune to it. Essentially, in the same way that if you get the flu one year, you don't get the same exact strain of the flu the next year. It's why, vaccines have to change every year for the flu. Which is very particular for the flu. I don't want to make it sound like there's a trillion coronaviruses running around. So yes, you can get immune. And actually, fun fact, that is how we're working on making things that will help against coronavirus for patients. So for example, we're taking antibodies, which is the immune response to it, from people who got better. And we're now trying to figure out ways to give them to healthcare workers, so then they won't get sick as they're helping people recover.
Great. Do we know yet if you can get the coronavirus more than once, this particular one that's affecting us right now?
It doesn't seem so. I know some people have been seeing probably cases of where it seems like someone tested negative and then they have it again. That's probably more of a testing issue. Our tests have relatively low what's called false negative rates, as in essentially saying someone is fine when they're actually not. But it does happen, which is what we think most of those are. Based on some information from SARS-CoV-1, we expect antibodies to last anywhere between 1 year and 3 years, just based on a related virus that people have seen.
Cool, thank you. Can viruses be used in a biomedical context? So are they ever useful for anything, or are they always bad?
They're amazing. Yeah. I mean, I've used them in my own research to turn genes on or off. They're awesome molecular tools, but they're not in a way that's like, this is not like engineering things in a lab for harm. This is more along the lines of like leveraging, using the capacity of the viruses to sort of infect cells so that I can tell my cells to turn on a gene or turn off of a gene. And I'm sure, Gabby, as a person who works in a full-on virology lab, you probably have more to elaborate.
Yeah, I love this question because viruses are actually the best tools we have. We learned so much about biology from studying viruses because they're such pared down, well, not really organisms, but things that still have to obey all of the laws of our cells and the way that they work, that we were able to essentially follow them through cells in order to discover that DNA is heritable, and things that were really, really important to biology like that. And actually, one of my favorite sort of virus fun facts is that there is an enzyme that we discovered in a group of viruses called retroviruses, like HIV, called reverse transcriptase, and it turns RNA back to DNA. I said before that DNA goes to RNA goes to protein. This just sort of switches it, at least at one step. And that's actually used right now in coronavirus testing. So in order for us to look at whether or not you have the virus, we amplify its RNA, but we first generally turn it into DNA, which is more stable and won't just degrade in water.
Great. So we've had a lot of people ask, how long do you predict it will take to develop a vaccine?
So vaccine production is quite a long time, because there's a bunch of steps that you have to take to first make sure that the vaccine is effective. Like, is it actually protecting against what we want it to protect against? And then B, is it safe for people to take, right? And so you have to go through a lot of testing in a laboratory to start making it. And then you have to push it through a bunch of phases of clinical trials so that you can make sure that it is both safe and it is both effective. And this is a matter of also statistics, right? We need to have statistics is a really important piece of math that we need to always keep in our biology work. And statistics allows us to sort of understand, like, are the results that we are getting, can they be extended to a large amount of people? Like, are we going to be able to the same result that we can say, okay, this is working really well in this lab situation, and there's enough people here that where it's effective and it's safe, that we can we can predict how it'll act outside of laboratory settings, right? And so usually they say that this process can be over a year, but there's a lot of resources and a lot of collaborations taking place right now, where that process is probably going to be sped up a tiny little bit. But I don't know. I think maybe we're looking at like 8 months to maybe like 16 months or something like that. Gabby, what are the ranges that you're hearing?
That's very similar, actually. I believe there's one didn't one make it to phase 2?
Yeah, so there is this amazing scientist over on the West Coast. I'll have to look up her name, but she was on CNN recently with Anderson Cooper. She's working on it pretty great with her group. I also want to point out that science never happens by only a single individual. So there are many people working on this. Science is a very collaborative. People have to work on teams, and they have to like share knowledge and share data. And so I think the cool thing that's coming about all of this research is that science is super global and it's super collaborative, and knowledge is definitely power in these cases. And so they're working on it.
And I also want to point out that it's not a vaccine or bust. We are also developing a ton of other things that can work to treat this that aren't vaccines. While a vaccine would be the final nail in the coffin and probably lead to this thing eventually going extinct, there are tons of ways that we can still treat it, and those are also being pursued. So there's something called antivirals, which block some step that the virus needs to do. And so there's, I think, one also in like phase 2 trials being tested. It wasn't even created for coronavirus, it was created for something else. And they realized, oh, this might work. And so there's a ton of other things that we're trying, essentially trying to repurpose medicines that might have been used for other purposes and test, are they effective? Do they help? And then there also is that sort of antibody therapy that I mentioned, of just giving antibodies that we know are good to someone else to at least prevent them from getting infected, which is very important for our healthcare workers who are exposed to the most number of sick people.
And then if I can just add, we also have this amazing cure in ourselves, right? Most of our immune systems are going to be able to handle the SARS-CoV-2 infection if we're getting it, right? And the only reason that we are it's not that we think everybody is going to get very sick from this. And I just also want to remind you out there, it is not your job to worry about this. It is the job of the scientists and the clinicians and our public health officials to make sure everybody is staying safe. And our only job is literally to stay in our houses and watch Netflix, right? And so it's not a bad lift for us. And so it is not your job to worry about it, and know that most of our immune systems are capable of taking care of this virus, either with no symptoms or maybe a couple of sniffles or things like that, especially in little kids. Little kids are not getting sick from this, so do not not worry about that. And you know, once we get our immune systems sort of figured out, and once we test how many people have already had the coronavirus, and then we get the vaccine into play, we have the antivirals, and then we also have antibody therapies. I mean, we are hitting this from so many different angles. It is the most amazing coming together of science I have ever witnessed in my life. I know it's a hard time for everybody, but it's actually a very beautiful time for science and knowledge being shared. And it's so beautiful. Our societies are coming together in really amazing ways.
That's very uplifting. Okay, so how many viruses exist that affect humans, ballpark? And then how many coronaviruses have we found that affect humans?
Gabby, I'll kick this to you since you're an evolutionary virologist.
Yeah, so there are so many viruses. I think predominantly humans get affected by fewer of them compared to other species. Some of that might just be the fact that we're very clean. We don't live out in the wild, which is kind of nice, so there aren't as many of them. I don't feel comfortable giving you a ballpark, but there's a lot. Like what Jeanne was saying about, you know, how many viruses, there's like 1,000 times more weight in viruses in the world than there are weight in elephants. There's a lot of different variations of viruses. The vast majority of them are against bacteria, so they're fighting a war we can't even see. But yeah, there are a lot of them. Coronaviruses, I believe that there are 4 that cause the common cold, and then that there are the newer ones, SARS-CoV-1, MERS, and then SARS-CoV-2. And SARS and MERS, I believe, are both extinct because they were a lot worse. And we were able to really like isolate it, identify it as a problem. This one snuck up on us a little bit more, but is also fortunately not as bad.
Yeah, and we have something in our bodies called the virome. So we have usually the average person has 12 viruses living inside of us at all time. And these are viruses that are not at all like impacting our health, right? And they're just like it's just like a nice little home for them. They get to hang out with like all of the bacteria that live inside of our bodies. They get to hang out with our cells. And so overall, like scientists are still studying this really cool phenomenon called the virome, which is basically a collection of all of the genetic materials housed by viruses that is inside of our body. And we're using that information to learn more about the interaction between human cells and virus particles, because one of the things I don't know if we said this, but most people do not think that viruses are alive. And so it's kind of an interesting way to think about viruses being inside of us, like what are they even doing there, and how are they interacting with our cells? So it's a really cool question, and more information will be coming out as people continue to study. And now that we have viruses as like the primary kind of focus of the world, I'm sure there'll be more funding for virus research, so we'll have more information about that coming up.
Hopefully.
Yeah.
Great.
Keep us with jobs.
It's always good. How do you get tested for COVID-19?
So there's two ways that you can think about testing, right? You can think about testing of an individual that is currently infected. So somebody who's presenting with symptoms like, you know, the major symptoms are like a dry cough, and a fever. And so if somebody has those symptoms, they can go to their doctor, and the doctor can take a little giant Q-tip and shove it up their nose and get some boogers and mucus and stuff like that. And then they take that swab and they isolate the genetic material that would be associated with all the goobers that are on that swab, right? And so then they look at the genetic material, so they do a genetic test, and that tells somebody if they have COVID in the moment, right? And then the second way of testing is after somebody clears the infection out of their body, they have antibodies that come up, as Gabby was saying, where, you know, these antibodies can stay, these memory antibodies can stay inside of our bodies for like a couple of years, and that protects us against future infection. And they can also test for the presence of those memory antibodies that are very specific against SARS-CoV-2. And I'm very excited about our friends at Mount Sinai who just got FDA approval for their antibody test. And so that's really exciting news for humans.
Great. So what are the benefits of wearing a mask, and should we be wearing masks all the time?
Do you want me to take this one? So,
Sure.
One, first, I would say try to avoid taking masks that are used specifically for healthcare workers. Things like you'll probably see surgical masks. Those are most important right now going to the people who really need them every day to deal with just tons and tons of COVID patients. But doing something as simple as tying a bandana around your face can actually be fairly helpful. Think about it, if you're coughing, that's going to trap the cough. And most people right now are wearing them, especially, so I live in New York City, which is the epicenter, and we have to wear them. I think they just made it like a
Yeah,
requirement.
there's some sort of like, you know, like request from our health department to do this.
Yeah, and it'll just basically catch some stuff that you're putting out. So it prevents, essentially, if you sneeze or cough, virus from being sprayed out so far into the environment. Because a sneeze can go really far, much farther than most people think. This is important too because, you know, there are some people who might be what we call asymptomatic, meaning you have the virus and you're producing it, but you don't show any symptoms. So people like my age are especially, you know, prone to this maybe happening. So even though I don't have any symptoms, if I go out, I'm still going to wear a mask because, you know, I haven't been tested. I don't know if I might be producing virus silently, so I don't want to sneeze it onto someone who's just trying to go and get some vegetables at the grocery store.
Yeah, and I want to add to this that I just want to make sure it's very clear that wearing a mask doesn't actually protect you against getting the virus. It helps to protect others in case you have the virus, right? So don't go out thinking that you're going to be able to wear like some bandana and you can then go hang out with your friends. That is not at all what's happening. And in terms of protecting others, it doesn't fully protect others, especially if it's a bandana. And remember how we talked about how tiny the viruses are, right? And even there's little water droplets that come out of our mouths even as we speak, right? And we can't see them. It doesn't, you know, it's not even a cough. And those virus particles are so tiny that they could still be in those little droplets and sort of go through the threads of the bandana, right? So it's really just a little tiny barrier to help prevent how much is going out there, but know that things are still getting through the bandana, even if you layer it a couple of times, right? So this is not the fail-safe, and you should wear it outside if that's recommended in your neighborhood, but it is not protecting you, so don't do activities that you would otherwise not do if you didn't have a mask on. It's not a protecting against you, it's just a protecting for other people.
Thanks. Could you explain basically how vaccines work?
Sure. Do you want to take this, Gabby, since I answered the last one? Or do you want me?
Sure. So vaccines, there's a bunch of different ways, depending on what specifically is in the vaccine, you know, how it works. But more or less what happens is in the vaccine, there is maybe a piece of a virus. So for coronavirus, it would most likely be that sort of outside portion of it that binds to our cells. And when that gets into our body, we have white blood cells, immune cells that can recognize it. And so what'll happen is they'll take that up, process it, you know, flag it, be like, hey, this is weird. And then they start producing antibodies against it. So it's kind of a complicated process with how the cells talk to each other, and then they start this long relay of making a bunch of cells that all, you know, express a targeting antibody against it. But when you get a vaccine, you get sort of a piece of the virus. It's kind of like getting a wanted poster inside your cells. And then all of your cells are now on high alert. And so if you see that, then all of a sudden, you know, your cellular police are ready to take that virus down.
It's like a really good training for your immune system. Like,
yeah.
it's like your immune system is like a superstar athlete, and this is like sort of preparation for the big race.
I've got an awesome question here. What is the difference between giving someone the memory antibodies from somebody else and the vaccine? Like, what's the difference there long term?
Cool. So giving someone antibodies is inherently more short term. So antibodies from someone else can only last around 2 or 3 months because you're not giving them the cells that actually make it. So although we can make like antibody factories in a dish in a lab and then use that to start giving it to a bunch of people, your body itself isn't the antibody factory in that case. When you give someone a vaccine, then you yourself are the antibody factory. And then there are some vaccines that, can give you protection for like upwards of 10 years. So that would be ideal for this, is that, once we've been giving people antibodies for the short term, then we have a vaccine that's going to last, much, 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 much longer.
Great. Next question from Vivi. I heard that a tiger caught the coronavirus. Are there other animals that we know of that can get coronavirus?
So yeah, they have shown some really interesting data on cats. So cats can get it from us, but we cannot get it from cats. Apparently that is the directionality of it. And has there been any research to suggest otherwise, Gabby, on that? I don't think so, right?
That's something I haven't been following as much,
Yeah.
But I know what's kind of neat is that it turns out ferrets can get infected with it, and we're using ferrets now in our vaccine studies because mice can't really get infected with it, and we rely heavily on lab mice normally. So when this first came out, everyone was like, oh goodness, what are we going to do? And then someone made the lucky discovery of ferrets, shared it with the whole world, and then now everyone's using ferrets. So that's another animal, and that's where it's helpful.
I'm very happy to say it cannot get into dogs.
It cannot.
Yeah, very, very good for Willow, my dog.
All right. There's so many questions here. It's hard to choose. What virus has caused the most deaths in humans in the history of time?
So for that, I can't think of one off the top of my head, but it's probably going to be a lot older. So my bet would probably be smallpox, based on the introduction in North America and like the Americas. So the thing about viruses is that a lot of them have been with us for a very, very, very long time. And so there were viruses that essentially many, many generations of people were always getting. Smallpox was one of these. It was relatively deadly, pretty contagious, much more contagious than this current virus that we are all stuck inside because of. But what I think really would have ramped up how many people it affected was that it predominantly was affecting people in Europe, and sort of that side of the ocean. And then people brought it deliberately to the other side because they knew that other people wouldn't be immune to it. They would have never seen it before. And so it really dramatically affected people who were living in North and South America who had never seen this virus before. And it unfortunately killed a lot of them.
Yeah, and also there's this really interesting visualization that you can look at called, it's a infographic on the visualizing the history of pandemics. Now, not every pandemic is caused by a virus, just FYI, but so smallpox had killed 56 million people. And that was in the 1500s when that big thing took place. But the Spanish flu, which the pandemic that took place in 1918 to 1919, took the lives of up to 50 million people. So that was pretty big. And then we can't forget the AIDS HIV pandemic, which also was something that we have to be very careful about.
Yes. Is COVID-19 more infectious than the flu?
I think it's slightly more infectious. So in virology, we have this concept called R naught, like R with a little zero below it, basically. And what that means is it's sort of how fast the virus replicates. How many people does the average infected person make sick? So for the flu, I think it's around 2 people. It might also be that low in part because so many of us get the flu shot every year that it at least helps, even if it's not exactly the same strain of flu that you're getting the next year. I think coronavirus, it's probably closer to 3 or 4, but we don't have a good handle on it because we're only really testing people that we expect to have gotten it. After this is over, we're probably going to be randomly sampling the population and looking for, even people who don't think they've been exposed to it. Well, do you have antibodies against it? Were you actually exposed to it? It is not, like I was saying earlier, the most infectious virus we've ever seen. So for example, measles has an R0, a reproductive rate of 12. So measles is extremely contagious, which is why we all get vaccinated against it. This one is not that bad.
Cool. All right, so we try to keep these at 45 minutes, and we've already gone over because we had so many questions, and I wanted to get everybody's questions answered. But we always try to answer, or end rather, with the same 2 questions for everybody. The first being, what's something that you wish everybody knew that's related to your area of expertise? And the second question is, what's something that you wish everybody knew about literally anything? It can be as silly or significant as you'd like.
So I can start with what I wish everybody knew, in that is, and I think we're seeing this in the research that's taking place right now, that science takes place through a process, and that we need things like failure and learning from the failure to keep, it's a cycle of failure and repeating it with like little adjustments. And that science takes time, and that science is done by people. And that's really what I hope people can glean from a lot of what this is all about. And then regarding what I hope people want to know in general, I think for me, I love the idea of people understanding that science is a part of our everyday.
You can still see me, Erin, and hear me, right? Gabby, can you hear us? Oh, Gabby's back.
Yep, sorry.
Okay, I don't know what happened to Jeannie. We lost Jeannie, but do you want to answer that question?
What was, the one thing you wish everyone knew about your field, and then one thing you wish everyone knew in general?
Yep.
Well, I don't know if this would count as in general or my field, but science is not always the same as English, which I think sometimes unfortunately gets lost in translation. And so I think sometimes it leads to people accidentally not trusting science or misinterpreting what scientists are saying, when in reality it's just sort of a language block. So for example, you'll hear things about, well, there's no scientific evidence that blank. It doesn't mean we haven't found it yet. It means that we've really, really, really been looking. And so just little like language tweaks like that are sort of more of a quirk of science itself. So we also say like, this suggests or this supports, rather than saying this proves, because in science, we're always looking for more information to support something. We never want to just assert that it's true. We have a saying in science, never say always, which is very helpful because biology is weird and it's wacky, and there might always be one tiny exception to something somewhere. So I kind of wish more people would not be looking for loopholes in what scientists are saying or thinking that we're being tricky. It just happens that we're speaking a slightly different language. I might have to make a science to English translation dictionary one of these days. What's another one? I guess about viruses specifically. Viruses are really fun. This goes back into that really great question we had earlier about, are viruses all bad? Viruses are actually really, really, really important. Every 2 days, half of all of the bacteria on Earth die from viruses. They're really important in making sure that our ecosystems work well, that our guts and all of the bacteria that we rely on to help us digest our food are kept in check and don't just grow like crazy. So while right now is an example of a virus that's unfortunately keeping us all indoors and it's not really that great, viruses are actually very important, and writing them off as all evil or terrible is not always really what we want to do. And it's a cool place to start investigating just science in general, I think.
Great. Okay. Well, thank you so much for joining us. I'll send Jeannie an email saying thank you that she joined us too. And Erin, thank you for signing for us. And thank you all for joining us and learning. I'm sorry we didn't get to all of your questions, but where can we find you all on social media?
So I know Jeannie has a Twitter. I do not. Oh, did she come back?
There she is. Jeannie, where can we find you on Twitter?
Sorry about that. You can find me on Twitter at JeanneGarb for my personal Twitter, and you can also find RockEDU at ROCKEDU_.
Great. Okay. And so, yeah, that's going to wrap up today. We're going to be talking on Monday about insects. That will be at 1 p.m. Eastern. On Tuesday at 2 p.m. Eastern, we're going to be talking about viruses again, and a little bit different viruses, not specifically about COVID-19, but if you had questions here that didn't get answered about viruses in general, come on over. On Wednesday, at 1 p.m. Eastern, we're talking about sharks with Jasmine Graham. And then Thursday, we're talking about how space exploration has kind of prepared us for social distancing. That's going to be pretty cool. And then Friday, we're talking with the phenomenal scientist, Kat Milligan-Meyer. She's going to be talking with us about microbes. Kat is just 10 out of 10 amazing. So I'm really, really excited for that one. So not that the other scientists aren't also amazing. I'm kind of like a friend of Kat. So at this point, we're going to say goodbye. Thank you for joining us again. If you can support us on patreon.com/SkypeAScientist, we'd really appreciate it. It helps keep us functional. And with that, we'll see you next week. Thank you again, Erin, Gabby, and Jeannie.
Thank you.
Bye.
Bye.